So we have taken this path because it is known that CAR T-cells are not very effective. So to try combinations with CAR T-cells. And one of the approaches that we took, both ex vivo in primary culture models of CLL lymph node samples, but also in a mouse model of CLL, the iwTCL1 mouse model, we combined both therapies, bispecific antibodies plus CAR T-cells. And we have seen tremendous efficacy of this combination. So what we did is we started out with one dose of CAR T-cell administration to the mice and then waited a week so the CAR T-cells got active. But then by adding on the bispecific antibodies, we really boosted this response even stronger. So which led then to a cure of almost all the majority of the mice, which was something I've never seen in this mouse model. So I believe that combining these two strong immunotherapy approaches would be a great benefit also for patients.